Heather Best is a scientist, researcher, about to submit her PhD next month, hopefully soon to be Dr. Heather Best. She's been the very lucky recipient of a MDNSW Research PhD Scholarship, which was made possible because of the Sue Connor bequest, which has been an amazing resource for our organisation and for the scientists who benefited from it. I think Heather might even be the fourth recipient from that bequest, which is amazing. And every year we invite the recipient of that bequest to come and tell us about their research. So Heather's doing her PhD at the Institute for Neuroscience and Muscle Research at the Children's Hospital at Westmead.
Thank you for giving me the opportunity to speak today, and I'm really looking forward to sharing what the Sue Connor bequest has enabled for us. So my PhD has focused around identifying the genetic causes of muscle disease to determine new disease mechanisms to target for treatment. Historically, the muscle disease is diagnosed predominantly on how the patient presents symptomatically in the clinic. And then the subtype of myopathy or muscle disease is determined on how the muscle biopsy looks like under the microscope. And here is an example of healthy muscle, and we see the individual muscle cells with the nucleus where the DNA hub of the cell around the edges. And in centronuclear myopathy, the nuclei move towards the center of the cell, and this is required for the diagnosis of centronuclear myopathy. The different pathological endpoints associated with different myopathies are also linked with different genes, but historically, identifying the precise genetic diagnosis has been a huge odyssey. However, this is immensely important to allow the correct course of clinical care for the patient, and this is particularly important if it's known that there's a cardiac involvement so that this can be closely monitored. It opens up family planning options, and it allows insight into the disease mechanism, and from here we can look into novel treatments. So in the last 20 years or so, there's been a huge effort in trying to identify all the disease genes for myopathy, so any genes that are associated with muscle disease. And you might notice that the amount of disease genes has rapidly increased after 2003, which is when the human genome was sequenced. And huge technological advances in genomics has increased the number of disease genes to over 300, and now we are able to get a genetic diagnosis to over 50% of our patients. However, moving forward, we want to be able to provide a genetic diagnosis to all of our patients and know all of the genetic causes of muscle disease. To this end, the INMR, where I'm carrying out my research, the Institute of Neuroscience and Muscle Research, we collaborate with the Broad Institute at Harvard and MIT. And in the INMR, over a number of years, we've established a cohort of patients who have muscle disease but no genetic diagnosis, despite having gone through all of the golden standard diagnostic tools that are available to us at the moment. So we send the DNA to the Broad, and they use the most up-to-date sequencing technologies to identify the precise genetic variant of these patients. One such cohort utilized whole exome sequencing, and this is where we look at just the section of the genome which encodes proteins, and this takes about 1%, makes up about 1% of the genome. The types of variants that came back from this particular cohort included new variants in genes that were known to be associated with muscle disease, and often these had a different clinical phenotype, and this is really important for the clinician to be able to make future diagnoses. And in 8 cases, we identified new genes that had previously not been associated with muscle disease, one of which I'll be sharing the odyssey of today. The Broad Institute identified the first variants with whole exome sequencing in a gene pyroxD1. And the index case was in Sydney. They came into the Children's Hospital at Westmead, and there were 2 brothers who presented in early childhood with muscle weakness and generalized reduction in muscle bulk. They had facial weakness, and this had some unique phenotypes, which was difficulty swallowing, feeding, and a unique nasal voice. So we didn't know how the gene PYROXD1 was associated with muscle. So we typed it into PubMed, which is a search engine for scientific articles, and we found that no paper had actually been published on PYROXD1, so nothing was known. So we really had our work cut out for us to establish PYROXD1 as a new disease gene for muscle disease. So we took a multidisciplinary approach with this. We utilized the unique clinical and pathological features of the patients and bioinformatics, as I mentioned. And then we used model organisms. We used yeast, which is highly versatile in the lab. We can manipulate it to do a range of experiments, and we used this to identify what PYROXD1 did. And then zebrafish is also a very versatile organism, and it's commonly used to make disease models of genetic conditions. And this is one step up from yeast because we're able to carry out assays to mimic the muscle dysfunction in our patients. So the multidisciplinary approach that we've taken went outside the scope of our lab, and at the end of this project, there were actually contributions from 42 individuals across 18 institutes across 5 countries. And this isn't the normal number of people that are on a paper, but this is becoming the norm when you're establishing new disease genes and rare conditions. So it was amazing to be part of this collaborative effort. So I mentioned the unique clinical presentation, and this was coupled with a unique histological presentation. I'd mentioned that particular pathological endpoints were linked to particular genes. And in this case, we see 4 pathological endpoints from 4 different myopathies in the same biopsy, which has never been seen before. So here we see internalized nuclei and variation in fiber size, which is typical of centronuclear myopathy. When we apply an oxidative stain to the muscle, we see areas absent for the staining, and this is typical of areas absent of mitochondria, which is the powerhouse of the cell creating our energy. And this is typical of central core disease. And we see aggregates within the muscle that have the same proteins that are present in myofibrillar myopathy. And when we zoom into the muscle biopsy with electron microscopy, we can see aggregates that are typical of Nemaline myopathy. So we were able to use the unique clinical and pathological features to spread the word to our collaborators all over the globe, and we came back with 5 families, 9 individuals in total, that presented with PYROXD1 myopathy and had variants within the PYROXD1 gene. We then wanted to find out, is the PYROXD1 protein, is it actually present within the muscle cell, and if so, where? So here I'll be showing a muscle biopsy, and if you imagine the muscle cells as long pieces of spaghetti, and we're looking at the spaghetti on its side. And that means we can see the sarcomere, which is the portion of the cell that contracts, so allows the muscle to contract. So to orientate ourselves within the biopsy to track down the cells, I first stained for the nuclei, as we can see in blue. And then to orientate myself within the cell, I've labeled the edges of the sarcomere, the contractile apparatus of the cell, and you'll see these in green. And then I probed with PYROXD1, which will come up in red. And we noticed 2 things. We noticed that PYROXD1 was present within the nucleus, where our DNA is, and it was present within the sarcomere. However, it looked like PYROXD1 was a protein on the move, because while we saw it in the nucleus, it wasn't present in every single cell. And even though it was in the sarcomere, where the contraction occurs, we noticed different patterns. Here we can see it in the center of the sarcomere, and in this image we can see it in the eye band near the edges of the sarcomere. Next, I wanted to find if the localization within the muscle cells differed in our patients. So here I will be showing a muscle biopsy again, but then it's as if I've cut the spaghetti and I've turned it and you're looking at it down the side. So again, I've orientated myself with the nucleus, and you can at first notice that there's a dramatic increase in the number of nuclei in the patients, which is very typical. And then when I stain with PYROXD1, I find that we have a reduced amount of pyroxD1 in the sick fibers. So you'll notice that there are some splitting, so the cells are breaking down, and that the nuclei have moved into the center of the cell. I also noticed that while in the cell itself we don't have as much pyroxD1, it seems to have moved within to the nucleus, and this made us wonder if the role of pyroxD1 was fundamental to our DNA. So our first step to identifying what pyroxD1 did was evolutionary analysis. So we looked at the pyroxD1 sequence and we saw which genes this matched most closely to. And so we found its close relatives. And so these are the genetic cousins of pyroxD1, and it resided in the pyridine nucleotide oxidoreductase family. And this is a family of enzymes, and they reduce thiols. And so this meant that the predicted function of pyroxD1 was also to reduce oxidized thiols. And this is really important for cellular processes because the redox balance has to be right for cellular function. For certain processes to occur, oxidation occurs, and if this can't be reduced by redox, these processes are effectively stuck, so the cell is sick because particular processes are frozen. So our collaborators in France developed a unique assay in yeast, and this is the budding yeast Saccharomyces cerevisiae. And they altered the yeast slightly, so it doesn't have glutathione reductase, and this is the major pyridine nucleotide oxidoreductase within yeast, which effectively means it can't deal with oxidative stress. And the assay that they undertook was to throw hydrogen peroxide, which gives a huge amount of oxidative stress to the cell, and see if it could survive. And in this case, it cannot. And we wanted to see if pyroxD1, which was meant to reduce the thiols, so rescue the cell, if this was the function. And we found that when we added human pyroxD1, we found that the cell was able to survive under oxidative distress. So this means that pyroxD1 does reduce thiols. And then we wanted to find out that do the patient variants, are they still able to reduce thiols? And we found that this was not the case. And so this lent evidence that pyroxD1 was the cause of the muscle disease in these patients. We then wanted to generate a disease model to confirm that pyroxD1 was the cause of muscle disease in these patients. So our collaborators in Melbourne generated a zebrafish where pyroxD1 is substantially reduced throughout the skeletal muscle. And the assay that they do to determine if the zebrafish has muscle weakness is effectively they tap the zebrafish and see how quickly it swims away. And they showed that when the pyroxD1 was lacking, the swimming abilities was markedly reduced. And when we look at the muscle, we see the same features that we see in our patients. We see wasting of the muscle cell, and we see the specific aggregates that we see within our patient muscle. So after 6 years of research, we had established pyroxD1 as a new disease gene for muscle disease. And now if you type pyroxD1 into PubMed, there will be a single hit. So not only had we identified a new gene linked to muscle disease, we had characterized an entire new gene. And importantly, we had shown that different pathological endpoints seen in a huge range of myopathies were linked to oxidative distress. And so we believe that we've found a core pathway for all of the myopathies that can be targeted for treatment. So we think that investigating further into pyroxD1 and pyroxD1 myopathy will be the key to determine how the oxidative distress is causing muscle disease. And oxidative distress is a very common feature of many other diseases. And the wasting of muscle cells, areas lacking in the mitochondria, and changes in cell size are very typical of motor neuron disease. And this raises the possibility that treatments that are being targeted for neurodegenerative diseases based around antioxidant therapies may actually also be able to use for not just pyroxD1 myopathy, but for a range of myopathies. And the link to neurodegeneration and nerves is strengthened because some mouse studies that I've undertaken, I do see PYROXD1 is expressed within the brain. And our oldest PYROXD1 patient is beginning to present with nerve symptoms. So he has mild neuropathy in the peripheral. And I've looked at other cells of the patient. So I've got skin cells, and PYROXD1 is there, but there's no phenotype associated with it. They don't have any abnormalities in their skin, and the cells are perfectly fine. So it looks like the distress caused by lack of PYROXD1 function is actually just in long-lived cells. So we want to develop a model that we could further investigate PYROXD1 disease, and one that very closely mimicked it so that we can test any treatments on this model. And I've developed a mouse model where the mouse is lacking PYROXD1 just in the skeletal muscle. And once we verify that this mouse does present with a myopathy that's similar to PYROXD1 myopathy, we want to see if treatments that reduce thiols, so this is what PYROXD1 does, we want to see if these drugs will be able to take on the function of PYROXD1 and if it will be able to cure PYROXD1 myopathy. During this process, I found that if you globally knock out PYROXD1, so when PYROXD1 isn't present in any cell in the body, the mouse doesn't survive. It's actually not born. And studies since then in single cells where they've taken one cell and removed PYROXD1, the cell dies. So we know that PYROXD1 is essential for life. And this leads on to one of our other predominant research questions undertaken from a postdoctoral fellow in our lab, Frances Everson. And she's looking into what does PYROXD1 reduce that is essential for life and also links to a huge range of the myopathies. So we've identified variants within PYROXD1 as a new genetic cause of muscle disease, and this gene has been previously unstudied. We've shown that the pathological endpoints of a large range of myopathies are ultimately linked to oxidative distress. And we believe that this is a core mechanism of disease through a range of myopathies, and it's also linked to motor neuron disease. And this new mechanism means that we can utilize any treatments that are being targeted for neurodegenerative diseases for children with rare muscle diseases. Thank you again to everyone who's made this possible, and of course our funding bodies. Thank you for listening, and I'll take questions.
Anybody have any questions for Heather? Joan's got a microphone.
Sorry, I'll try and catch up. So, the PYROXD1,
I'm trying to help.
that was just across a certain type of muscular disease.
Yeah, that's right.
So there's lots of different ones here.
Mm-hmm.
So it could apply to some of the other muscular dystrophies.
Yeah.
We don't know that yet. Or
So we think that the mechanism of the breakdown of the muscle in patients that have PYROXD1 myopathy is caused by oxidative distress, and we think that that this is actually a core mechanism across a whole lot of myopathies. It's when the cell has too much oxidation, so effectively you have a lot of processes within the cell that can't take place. You mean like apart from the lack of PYROXD1?
See, in this case, it's caused by a lack of this enzyme that's part of that pathway. But we're all subject to oxidative stress. And in fact, there are some people who believe we should all take antioxidants. So you can buy antioxidants. This is going to be my question. You know, you can buy antioxidants in the chemist. There is a logic. There has always been a logic, but maybe even more so with this for all of us, particularly with muscle problems, taking antioxidants.
I personally think that that's absolutely true.
Yeah. So, you know, vitamin C, vitamin E, coenzyme Q, blueberries, There are lots of antioxidants in our diet.
And conveniently red wine and dark chocolate.
So whilst Pyrox D1 is not a public health problem, I think you could say, it's hopefully giving us a clue to the common pathway or common endpoint of lots of different muscle conditions.
So how can you improve the Pyrox D1, or does adding that help with the disease?
So I guess we're taking a slightly different approach. So we're not targeting Pyrox D1 directly. We're identified what it does, and we're trying to see if a drug can take on that function.
That will take a long time, I presume, to get to that point.
Not necessarily, because there are drugs out there that reduce thiols that have been used in clinical trials for other diseases, and so that's what, once we show that our mouse model does have a myopathy, those are the drugs that we want to use, because we don't have to have the 10 years to get them through clinical approval. Everything's already set up, ready to go. Yeah, so that's the hope. Yeah.
So we're teetotalers, so we need to start on the red wine.
I advise that strongly. Yeah.
Is red wine better for you? Is it actually better than red grape juice? I mean, this is a totally non-medical question.
I just want to say yes. I don't know though.
Yeah, I'm just wondering whether it's propaganda by the alcohol company.
She's looking for permission.
Yeah.
Yeah, I give you permission.
As well, but I'm just wondering whether fruit juice is as good as alcohol made from fruit. It's not as fun.
Yeah.
Yeah, but that's not the question.
Yeah.
Let's keep moving. One more question? Is there one more question? And then we might move on. Yeah?
So, for years, they sort of, identified that taking folic acid when you're pregnant, reduces the possibility of, spinal bifida, etc. So for someone like, because we have a family history, I've got 2 girls, came from nowhere. If I've got a daughter that's trying to have a baby, and so we do folic acid and all that kind of thing, is it her taking on that antioxidant approach of the plant concept, is that going to make any difference? It's either there or it's not.
Yeah, I think that's a really good question. So one of our, we're interested to see if it would reverse the distress in the muscles, or if we do actually need to hit it before it happens. I think that the difficulty with that is that it may not actually go through the placental barrier, and that may be the issue. But I think that if we could target in the womb, then I think that is safely, then that would be a better approach.
Are you talking about other muscle diseases or Pyrox D1 deficiencies?
Well, no, ours is a
general, yeah.
very unknown.
I mean, I
Yeah.
think
At Broad Institute, still on an unknown. We
Yeah.
can talk before we're finished.
Great.
But I just sort of think she's at 33. She's considering her options. We haven't got a diagnosis. So if you're covering the bases to make sure this can't happen again in the next generation,
I suspect it's going to come down to
can't hurt.
Well, I was going to say, as long as it's safe.
Yeah.
But I said, if you had Pyrox D1 deficiency, the dose that you would need to make a difference is going to be very high. So you're sort of talking in 2 different levels. I think antioxidants are good generally, but lots of something isn't necessarily better than a little bit, and it's hard to know where to pitch. Okay. I think we might move on. Thank you, Heather. Thank you.